Okay. So can you hear me at the back? Yeah, okay. So this is just to put you into context. This is just an example cross-section from CT scan across the thigh of a healthy 20-year-old woman and a healthy 80-year-old woman. And the thing that people look at immediately is the loss of bone mineralisation that's occurring, but I also draw your attention to the dramatic loss of muscle mass that has occurred over the period of time. If we put a little bit of descriptive science on that, this is work, really quite amazing work from Jan Lexell, which went on in Sweden a number of years ago. And what we're showing here is data from accident victims where they were taking full cross-sections of the vastus lateralis muscle, and then they looked at the muscle cross-sectional area with age, and they looked at the numbers of muscle fibres within the muscle with age. And you can see that muscle mass is maintained roughly to about into late 40s, early 50s, and then declines. And the predominant reason why there's that decline is because of the loss of muscle fibres. When people have compared the loss of muscle fibres to information on motor neurons and on the innervating neurons, then what seems to occur is that there's a total loss of what's known as the motor unit. So the motor unit is the muscle fibre, it's the innervating motor neuron, and of course it's the anterior horn cells as well. And there's evidence that we actually lose anterior horn cells, we lose motor neurons and the associated muscle fibres, and then there's also some sprouting and re-innervation goes on in you and I as we age. The big unknown question here is whether the muscle goes first, the muscle fibres go first, or whether it's the nerve that goes first. And trying to understand that is one of the really difficult things that we're trying to do. Now, most of us, in fact almost everybody in this area, do tend to work on the muscle end of this particular system. But certainly at the end of my talk, I'll come back to sort of different ways of trying to approach which part of the motor unit is likely to be going first, or whether it all goes together and so on. So, like most people in this area, we study rodent models because it's a tractable system that we can study over the lifetime of the animals as such. And so what we've got here is just an example that over the lifetime of the mouse also loses maximum muscle force. So this is the extensor digitorum longus muscle of the mouse. And as well as actually losing fibres, the fibres that remain are also weak. And this is the specific force generation, so the force per cross-sectional unit area, from that muscle in the rodent as well. So we've been interested for quite a long time. We started being interested in whether the oxidative stress, the oxidative damage theory of ageing was something that we could find evidence for in our experimental models. And in more recent years, as you'll see, we've become much more interested in redox signaling and the possibility that changes in redox signaling are playing a crucial role in this loss of muscle fibres that we're seeing. And this is what I'm going to talk about. I'm going to tell you a bit about some of the older work which just indicates that in all tissues from aged animals and aged people you see evidence for oxidative damage. There's also evidence for increased oxygen generation. I'll talk very much about the area that we're interested in, is the way that skeletal muscle specifically adapts to stresses of contraction and other stresses by increasing reactive oxygen species generation, and they stimulate adaptive responses. And then I'll come back at the end to talk about our work with colleagues in the United States, in which we're trying to modify reactive oxygen species activities in muscle and nerve and look at how that affects muscle aging. So first of all, just one slide on oxidative damage. Like other people, if you take muscle, what you see is if you look at things like carbonyls, then you find an increase in protein carbonyls in tissues or muscle from old animals compared to young. If you look for oxidation products like malonaldehyde, they're increased in the old. And that's thought to be predominantly due to the fact that mitochondria from old animals are leaking or increased amounts of generation of reactive oxygen species, such as hydrogen peroxide. And these are data that Holly van Remmen did in collaboration with us, where we took muscle from old animals and young animals, we extracted mitochondria, and we looked at hydrogen peroxide generation, and you can see it's elevated. And that's really something that you can see in an awful lot of models if you look at aging tissues. But what about this second area? We know, and I'll show you some data in a moment which confirms the fact that when we contractile muscles, when we exercise, we generate reactive oxygen species. And it used to be thought that actually this was potentially something that we should, was very bad for us and we should worry about. And it was only really as we began to realise, and there's a number of people in the room who've done this work, that actually muscle in particular adapts very rapidly to those changes in reactive oxygen species generation, that we began to understand a bit more about what was going on. So over a number of years, we've been trying to identify the sites of reactive oxygen species and reactive nitrogen species generation in skeletal muscle. And this is one of the latest slides that we've got to look at that. So that most of the work in the past has been done looking at the mitochondria and the suggestion that during mitochondrial respiration, there's release of superoxide, or dislocation superoxide, the formation of hydrogen peroxide, and that hydrogen peroxide can potentially diffuse through the mitochondrial membranes and into the cytosol and so on. There's also evidence for various different cytosolic sites of superoxide generation. I'll come back to talk about those later on. And again, in the presence of superoxide dismutases and also with spontaneous dismutation, we get hydrogen peroxide formed. We've been very interested recently in whether any of that superoxide is reacting with nitric oxide, which is formed from nitric oxide synthases within the muscle to generate peroxynitrite. And then there's a series of generation systems or potential generation systems on the plasma membrane. And it's interesting that Dr. Murray will follow after me, because we've been particularly interested in whether there's a plasma membrane oxidoreductase system which is generating superoxide on the outside of the muscle cell. So there's a complex system. We know that some of those are activated during contractions. So during contractions, what happens? Why don't we get lots of oxidative damage? Well, we've studied what happens by looking at this in vivo and in vitro models. And our in vivo models, which originally came from John Faulkner's lab at the University of Michigan, we actually stimulate individual muscles using, of anaesthetised animals using stimulation of the motor nerve. And then we actually can take out the muscle which has been contracted with a certain protocol, and we can analyse the changes that have occurred in that. And if we do that and we look at what happens with a single period, single 15-minute period of isometric contractions in these animals, what we find is that very rapidly we get an upregulation of these protective enzymes, similar sorts of things that Dr. Risto was talking about earlier on. So the total superoxide dismutase rises very rapidly, about fourfold over 12 hours. The catalase rises over a longer period of time. But what was interesting when we did this work about 10 years ago now was actually we saw some really dramatic changes in heat shock protein content in the muscles. And so this is HSP60, the predominantly mitochondrial heat shock protein. As you can see, this is going up 1,500% with this activity of these muscles, and HSP70 was going up by about 400%. So the muscles are adapting very rapidly to a period of contractile activity. Various workers have looked at the mechanisms by which that occur. And of course, again, we've heard some from Dr. Risto already. We'll hear more, I'm sure, from Dr. Vigna in terms of some of the changes in transcription factors that occur. We've been particularly interested in HSF1 because of it's involved in the transcription of heat shock proteins, activator protein 1, and NF-kappa B. And all of these are activated by this short period of contractile activity. And there's evidence that reactive oxygen species, either directly or indirectly, are involved in the activation of these transcription factors. But what happens when we age? So let's just step back. So what's happening, seems to be happening in you or I, is that as we exercise, as we undertake contractile activity, there's an activation and generation of a variety of reactive oxygen species. If that's over a certain threshold, which we don't really understand, then there's an upregulation of protective systems. And that seems to be a very dynamic process, that as we actually exercise, we exercise regularly, it goes up and we get an increased protection. If in fact we stop exercise, then that down-regulates and so on. So it's a dynamic process. And in young and adult animals, young and adult people, that certainly seems to occur. What happens as we age? Well, if we try and do the same experiment in old animals compared to young animals, the first thing we have to ensure is that the muscles of the old animals are doing the same amount of work as the muscles of the young animals, and that's shown here. And then if we look at some of these products, in this case, another of these heat shock proteins, HSP25, what we find is that in young animals, following the exercise protocol, the 15-minute exercise protocol, we get a big upregulation of HSP25. In old animals, this is completely abolished. So we don't get this upregulation, we don't get this adaptation occurring to the contractile activity. If we then look back at some of the signalling pathways, in this case of the transcription factors which we think are involved, NF-kappa B, what we've got here is an EMSA then looking at NF-kappa B activation in adult animals and all animals, both pre and post-contractions. And so you can see in the adults, there's an activation of NF-kappa B following contractions. In the old, there's already evidence that NF-kappa B is activated at rest, and there's no further activation with contractions. And exactly the same occurs with AP1. So there's an inability of these signalling systems to be activated following the contractile activity. So is that important? Well, it was difficult to actually think how we were going to decide that was important. One of the ways that we've tried to approach that was to say, well, what are some of the key products of these signalling systems? And if we actually can identify them, say they're heat shock proteins, can we overcome that block in activation that occurs with aging? And the way we've done that, like many people, is actually to say, well, if we overexpress some of the products throughout life, will they protect against the loss of muscle mass that occurs or the increased muscle damage that occurs? And so we've done a whole series of these, but the one that's really looked interesting is HSP10. Now, HSP10 is a co-chaperone with HSP60, thought to be predominantly within mitochondria. And so this is work that we've done with Dr. Wolfgang Dillman at the University of California at San Diego. And these are animals which are overexpressing HSP10 in their muscle. And so what you can see here is this is the overexpression. The overexpression is maintained into old age. It has no effect on the expression of the other heat shock proteins. And the overexpressed HSP10 is predominantly within the mitochondria. The really dramatic thing is that this overexpression prevents the loss of muscle mass which occurs in these animals as they age. So what we've got here then is the maximum tetanic force generated by the muscles of adult animals, and this is the loss that occurs with aging between here and here, and this is the HSP10 overexpressing animals, and you can see that's completely prevented the loss of force. So in this experiment model, we've managed to prevent the age-related loss of muscle mass. How's it working? Well, it's reducing the amount of oxidative damage, certainly, that we can actually find in the muscles of the old animals, the old HSP10 overexpressing animals. And we thought, fine, so what it's going to do is actually prevent the increased oxygen generation by mitochondria. So, again, working with Holly van Remmen, we took we extracted mitochondria from the muscles of these animals, but in fact there was no change. So the HSP10 overexpression doesn't seem to be capable of preventing the increase in mitochondrial reactive oxygen species generation. What it does seem to be doing is changing the expression of the protease, which is particularly involved in clearing oxidative proteins from mitochondria. And Calvin, I think, is going to tell us about that, but it's known as the LON protease. So the LON protease seems to be elevated in the HSP10 overexpressors. So it looks like that this HSP10 overexpression is somehow or other influencing the ability to deal with oxidative damage in mitochondria of old animals. This is just one slide to actually show that we're actually trying to translate this into people as well. Does this failure of adaptation occur in people as well? And obviously it's much more difficult to do this sort of work in people. We can get muscle biopsies to do some aspects of it. But if you take lymphocytes from young people and old people and you stress them, you also find that there's this attenuation of the ability to generate stress proteins as well. So it looks like at least some aspects of it is also reflected in older people. So the final part of my talk then is to say, well, if reactive oxygen species are particularly important, can we find models that allow us to modify the rate of oxygen generation? And so we've worked this time with Alan Richardson and Holly van Remmen in San Antonio to actually look at animals which have modified regulators of reactive oxygen species. And so we've looked at manganese SOD knockouts, we've looked at GPX1 knockouts, we've looked at catalase knockouts, we've looked at catalase overexpressors. And very briefly, as you heard before, the animals which have become most interesting to us are animals which are knocked out for copper zinc superoxide dismutase. So the reason for that is actually if you then look at their muscle mass with increasing age, what happens is they've got a premature loss of muscle mass. So this is the wild-type animals up here. You can see it coming out to 28 months of age. But in the young copper zinc knockouts, what happens is that they maintain muscle mass initially, and then it falls off more rapidly. And we thought this was an interesting model to allow us to look at how the process by which that might be occurring. So what does copper zinc SOD do? Well, we all know that actually it's present in the cytosol and it's involved in the dismutation of superoxide to hydrogen peroxide. When we started this work, we thought that was its major function. But in fact, it turns out through work that Enrique Cardenas and others have done that a proportion of the copper zinc SOD is also sitting in the mitochondrial intermembrane space. In fact, probably about 10% of the total copper zinc SOD is sitting in the mitochondrial intermembrane space. And if you think about most cells, in fact, probably manganese SOD is only present at about 10% of the total SOD. So the copper zinc SOD, which is sitting in the intermembrane space, is actually a very component of the protective ability of the mitochondria. So the copper zinc SOD is sitting in both of these two sites, and indeed what we find is that there's some mitochondrial disruption occurs in this model. This is shown here. This is electron micrographs of wild type and a 14-month-old and 22-month-old wild-type animals and copper zinc SOD knockouts. And you can see there's the mitochondrial accumulation in the SOD knockout. So this copper zinc SOD sitting within the intermembrane space of the mitochondria seems to be functional, and knocking it out seems to influence the mitochondrial morphology at least. The other thing that it does is actually disrupts this link between the motor nerve and the muscle. And so this is some imaging of the neuromuscular junction. So this is wild-type animals here. This is the acetylcholine receptor, which is visualized here. This is the innervated nerve, and this is the merge, showing that most of the neuromuscular junctions are innervated. In the SOD1 nulls, the copper zinc nulls, you can see that there appears to be disruption of the acetylcholine receptor, the innervated nerve is disrupted, and a whole series of the neuromuscular junctions aren't properly innervated. And this mimics to a certain extent what we see with aging. So we've become interested in trying to understand what's going on in this model, because it certainly seems to show some of the overt changes that occur with aging. Being interested in the free radical end of things, we've been particularly interested in exactly what the knockout of copper zinc SOD is doing. In the aging area, most people who've been looking at SOD knockouts have been assuming that what happens is that as you actually knock out the SOD, you get an accumulation of superoxide, and that superoxide is thought to inhibit aconitase and have other effects. There's been very little discussion about other things that can go on. Now, we've been interested for a little while in what nitric oxide might be doing in this sort of process. And the reason for that is because actually this particular reaction of superoxide with nitric oxide is able to go at roughly 3 times the rate of the dismutation of superoxide to hydrogen peroxide catalyzed by superoxide dismutase. Now, most of the time, the amount of superoxide dismutase that we have in mitochondria or we have in the cell is such that actually this is a major product. But there's been very little consideration of what goes on when superoxide dismutase is knocked out. And we've wondered whether, in fact, the key thing that might be occurring is rather than an accumulation of superoxide, we're getting peroxynitride formed in larger amounts. And that's actually what's causing the changes that we're seeing in our muscle. The other thing that argues for that is actually, if you look at the literature in terms of nitric oxide standing concentrations, if there is such a thing compared to superoxide in cells, and roughly there'll be at least 100-fold, maybe 1,000-fold excess of nitric oxide in cells. So what we wanted to try and do was to monitor the superoxide. And so, like many other people, we've been trying to find probes which we can use in our muscle cells to allow us to look at superoxide. And the one that we've been spending most time on is dihydroethidium. Now, many of us have looked at ethidium fluorescence in different model systems over a number of years, but you'll be aware that more recently there's been a specific product identified, the 2-hydroxyethidium, which is a specific product of DHE reaction with superoxide, and the reactions are shown here. And so there's a need to be able to actually monitor 2-hydroxyethidium. Now, so these are data from our muscle cells in which we've undertaken the experiment in two ways. One is to load our individual single fibres from wild-type animals and animals which are knocked out for copper zinc superoxide dismutase with DHE, and then look at ethiddium fluorescence, but also to analyse them using HPLC and look at 2-hydroxyethiddium fluorescence as well. I'm afraid it's a little bit too light in here, but up the top here we've got fluorescent images which show that when you use DHE in a system like this, what happens is the DHE actually accumulates in the nuclei, because of course we all use ethiddium to visualise nuclei in cells. If we look at the ethiddium fluorescence, then what happens, really very surprisingly, we find no evidence for an increase in ethiddium fluorescence in the copper zinc nulls at rest. And if we measure 2-hydroxyethiddium, you can see we find no evidence for an increase in 2-hydroxyethiddium at rest. If we stimulate these fibres to make them contract, then what we see is that in the wild-type animals, we get an increase in ethiddium, we get an increase in 2-hydroxyethiddium. So this is presumably reflecting superoxide, which becomes more available during the contraction process in these fibres. But if we look at our copper zinc nulls, really contrary to what we expected, we find there's actually less ethiddium and no evidence for an increase in 2-hydroxyethiddium. Doesn't make any sense, does it? We've knocked them out for copper zinc null, but we find no evidence for an increase in superoxide. What seems to be going on is that all this extra nitric oxide which is sitting around is actually reacting with the superoxide very rapidly indeed, and we're getting peroxynitride generation. Now, it's very difficult to measure that. Certainly don't have a probe for that. But you can look at the 3-nitrotyrosenes as a measure of nitration of different proteins. This is actually looking at 3-nitrotyrosenes in a major muscle cytosolic cytosolic protein, carbonic anhydrase 3. And what we can see here is that in wild-type animals, pre- and post-exercise, we get suggestion of an increase in the 3-nitrotyrosenes. In our copper zinc nulls, we get a big increase at rest with no further increase with contraction. So this seems to be evidence that, in fact, what's happening here is that superoxide, we're getting a big increase in peroxynitride. And actually, I won't show the data, but if you look at nitric oxide, free nitric oxide levels using DAF, we find a decrease in the DAF in these copper zinc nulls. We've also gone on to look and see whether there's some changes in nitric oxide synthases. The answer is we can't see any evidence for that. These are the Western blots quantified over here. A very bright reviewer, and he may be in the room, suggested that one of the things we should look at is peroxyredoxin 5, because peroxyredoxin 5 is a peroxynitrite, has peroxynitrite reductase activities, and is known to be upregulated in situations where there's increased peroxynitrite formation. And sure enough, as you can see down here, peroxyredoxin 5 levels in the muscles of these animals are grossly elevated. So it looks like that the thing that's going on here is that the knockout is causing formation of peroxynitrite, and that's initiating a whole series of other things which lead to this accelerated muscle aging, accelerated loss of neuromuscular function that we're seeing in these animals. So that's really just coming back to what we think is going on here. The thing that we're really interested in is, if this is a good model, does it also influence the ability of the muscles to adapt to contractions? And the answer is yes. So this is now looking at the activation of NF-kappa B again in wild-type animals, heterozygous knockouts for copper zinc SOD and homozygous knockouts for copper zinc SOD. This is the activation with contractions that you saw before. And in the SOD nulls, you can see the NF-kappa B is already elevated, or the activation of NF-kappa B has already occurred at rest, and we get no further increase with contractions. So coming back to this then, the lack of copper zinc SOD seems to lead to changes in the whole motor unit, which is similar to those that we see during aging. What else can we do with that? Well, of course, we've now got a model which is tractable and we can start to play around with. And so what we're doing with Alan Richardson and Holly van Remmen is actually introducing a rescue procedure into this, so we can actually re-express copper zinc SOD in either the motor nerves or in the muscle, and look and see what this does to the system. I haven't got the data to show you here, but actually the answer seems to be it's all happening in the nerve, which for a muscle physiologist is a bad thing. Okay, so what do we think is going on? Why do we think this is actually occurring? This is a the hypothesis that we published in the journal Physiology earlier this year. So what seems to be happening during normal contractions is that there's an increase in local reactive oxygen species generation, leading to an increase in hydrogen peroxide in the cytosol. Most of the work in the past has suggested that this should be mitochondrial, and this is mitochondrial. But in fact, over the sort of period of time that we are looking at, which is normally only 10 or 15-minute periods of isometric contractions, we've got more and more evidence that probably it's an oxidase of some kind, which is acting as a signaling oxidase in response to the contraction process. But whatever is happening at that end, what seems to be occurring in the cytosol is we're getting local thiol oxidation with activation of these transcription factors. The activated transcription factor then translocates the nucleus where it activates transcription. This is actually, and that transcription is then, among other things, is activating increased levels of proteins which protect against oxidative damage, heat shock proteins, the superoxide dismutase 2, etc., etc. This is where it then impinges on the work that we were just hearing about in the elegant presentation from Dr. Jones, because actually what we're now talking about is oxidation in one compartment of the cell leading to changes in another. And as Dean has just presented to you, there's evidence that the thiol disulfide redox potential in the different parts of the cell are changing. So what we need to be able to measure is what's happening with contractions in the cytosol and what's happening in the nucleus, because in particular things like NF-kappa B require a reduced nucleus in order to retain their binding to the DNA. So in other words, if you've got an oxidized nucleus, then what happens is that you don't get transcription. So in order for this process to occur, you actually have to have an oxidation in the cytosol, but a reduction in the nucleus, or a reduced environment in the nucleus. So what's happening with aging? Well, this is the data that you've just seen. At rest, it looks to us as though, whether due to a primary or a secondary effect, there's increased hydrogen peroxide release from mitochondria. And that's already causing an oxidation within the cytosol. That oxidation in the cytosol provides this baseline activation of NF-kappa B and AP1 that you saw presented earlier on. And that leads to potentially a transcription of some of the NF-kappa B products. You know, we know that there's an increased inflammatory environment, for instance, during aging. Something or other prevents this occurring further when we contract the cells. So that's at rest. What happens with contractions? Well, we aren't seeing this additional transcription of cytoprotective proteins. We're trying to understand what that might be. It might be a lack of the signal. It's occurring here. It might be an over-oxidation within the cytosol. But it might also be that this increased hydrogen peroxide is causing an oxidized environment within the nucleus as well, and therefore preventing this further transcription occurring. And that's the hypothesis that we're trying to understand at the moment. But certainly if we actually overcome this by overexpressing some of these key proteins which play a cytoprotective role, what we find is that we can actually prevent the loss of muscle mass and force generation that occurs during aging. So we think that it's a process that's worthwhile continuing to look at. So just to conclude then, muscle from aged mice and all mammals have evidence of increased oxidative damage. We've shown you that skeletal muscle adapts rapidly to contractions. It trains, in other words. We've shown you that those redox-mediated adaptations are attenuated with aging, that muscles from aged mammals and those from the young copper zinc null mice don't adapt to this increased rust generation that occurs during contractions, and that we've seen that the copper zinc SOD causes an accelerated muscle aging phenotype. Just like to thank the people that we've worked with over this. This is Alan Richardson and Holly Van Remmen. When we started this work, we were working with John Faulkner, who's now retired, but it's Susan Brooks at the University of Michigan that we're working with at the moment. And then this is our lab in Liverpool. Anne McCardle, Aphrodite Vasilaki are the people who've done most of the work. And I thank you for your attention.
Thank you very much, Malcolm, for the very interesting talk. So it's open for discussion.
nice presentation. As you pointed out, there is some double signaling with nitric oxide and superoxide depending on conditions. And I found that the decrease in nitric oxide synthase that we found in aged animals is interesting because it suggests less inhibition of cytochrome-oxidase, less peroxynitride formation, maybe for a faster recovery of the damage induced by contraction. Will you comment on that?
Okay, so we were struck that this model, which everyone assumed would lead to an increase in superoxide, that there were a variety of other potential things that could occur. You may not get as much hydrogen peroxide in these models in response to contractions, just because there's not dismutation. You may actually get an increase in superoxide. You may actually get a reduction in nitric oxide available because it's been reacting with superoxide, and that's why we did that work, Alberto. In that model, we didn't find any changes in nitric oxide synthase expression, but we do find a decrease in available nitric oxide, and we don't understand whether that's physiologically important at this stage, but of course it may be. Maybe you'd like to comment.
Just a comment. Probably 10% of the world's population is too low in zinc, or maybe even higher, and another 10% or so may be too low in copper. So a lot of human variability might be affecting this.
Yeah, I mean, there are people, aren't there, who are looking at expression of these key enzymes, but also looking at the genetics of them as well. And there's some evidence that, for instance, GPX might influence aging.
Just a comment. Even if you don't find any difference in the expression of nitric oxide synthase, since it is so highly complex, regulated, I mean, if you have an oxidative environment, you can change the availability of nitric oxide.
Yeah.
So.
That's very much, and that's very much we're interested in. We've done the reverse experiment, which is take animals that are overexpressing neuronal nitric oxide synthase in muscle. And what we actually find there is, again, that there's increased peroxynitride generation and reduced superoxide availability. But in that model, the superoxide dismutase is all upregulated, presumably to try and maintain some sort of homeostasis for peroxynitride.
Terrific talk. The question I had was, the transgenic models typically lose the overexpression, like the thyroidoxin models. They lose the overexpression as the animals age. Has anybody put any of the antioxidant proteins on a promoter that would actually increase with age? Because certainly there's some proteins that will increase.
No, you're quite right. I don't think anyone's done the experiment that you just suggested, but actually those of us who are interested in this sort of approach to doing things, you have to be incredibly careful. So one of the HSP 70 overexpressors, people said that had no effect on aging at all. But actually when they eventually looked, the HSP 70 overexpression had gone because the promoter wasn't as effective with aging and such. But it's a real problem in this area.
Malcolm, I think with aging there is an increase in activity and content in stress also. Is it a kind of compensation?
Okay, so, yeah, I mean, in some situations, unfortunately not all experiments, in our hands at least, a whole series of the antioxidant enzymes do upregulate. So we find an increase in manganese SOD, we find in some systems copper zinc SOD and so on. And that seems to be an adaptation, or attempted adaptation to an increased oxidant generation, increased oxidant activity that occurs with aging. Yeah.